There is a number your doctor has never told you. Not your cholesterol, not your blood pressure, not your resting heart rate. Something deeper. A number encoded at the molecular level of your DNA that reveals how fast your body is actually aging. Scientists call it your biological age. And for millions of people, that number is running 10, 15, sometimes 30 years ahead of the date on their birth certificate. I know, because mine was one of them. At 43, my joints ached like I was 70. My skin had lost something. That quiet luminosity photographs don't lie about. My energy was borrowed, never real. Every morning felt like a negotiation with a body that had stopped cooperating. I saw specialists. I ran every test, and every single time I received the same verdict wrapped in different language. This is just aging. This is normal. But here's what not one of them ever asked me. What are you eating? Not in the way a nutritionist hands you a pamphlet, in the way a forensic scientist would ask it, because what I eventually discovered was not a dietary preference, it was a crime scene. And the evidence had been accumulating inside my cells for decades. Here is what the latest research in epigenetic biology now confirms. Certain foods do not merely make you unhealthy, they reprogram the biological clock embedded inside every cell of your body, accelerating a process called inflammaging, corrupting your collagen architecture, silencing the very genes responsible for cellular repair. And the most devastating part? These are not obscure ingredients. They are not rare compounds. They are in your kitchen right now. I removed 5 of them, completely, methodically. And within 18 months, my biological markers told a story that made my physician set down her pen and stare. But before I tell you which 5 foods they were, you need to understand what they were doing to your body at the molecular level. Because once you see it, you cannot unsee it, and you will never look at your plate the same way again. They had a name for it, buried deep in metabolic research literature. DOT, a process so gradual, so invisible, that most people never feel it happening until the damage has already been compounding for years. They called it cellular oxidative burden. And the first food on this list was quietly engineering it every single day. Refined seed oils, canola, soybean, sunflower. The oils pressed into nearly every packaged food, every restaurant fryer, every label designed to make you feel safe. I had consumed them for decades without a second thought. Most people have. Here's what that choice was actually doing inside my body. When these oils are extracted and heated, they undergo a chemical transformation called lipid peroxidation, a cascading molecular breakdown that generates compounds called aldehydes. Imagine pouring rust accelerant directly onto the load-bearing infrastructure of your cells. That is not hyperbole. That is the biochemistry. A landmark reanalysis published in the BMJ found that replacing saturated fats with linoleic acid-rich vegetable oils, the exact dietary switch we were told was heart-healthy, was associated with significantly higher rates of oxidative damage and mortality. We were sold a lie dressed in clinical language. Within 6 weeks of eliminating these oils entirely, my inflammatory blood markers dropped. The brain fog I had normalized as just how I function began to lift. But oxidation was only the opening act. The second invisible assassin was sweeter, far more seductive. And the damage it was inflicting had a name that sounds almost harmless: glycation. Every time you consume refined sugar, white bread, pastries, sweetened drinks, the glucose surge flooding your bloodstream triggers a process where sugar molecules physically bond to your proteins and DNA. They corrupt, they cross-link, they form what biochemists call advanced glycation end products. AGEs. The acronym is not accidental. Picture your collagen, the structural scaffolding that keeps your skin firm, your arteries elastic, your joints fluid, as a vast suspension bridge. Glycation is the rust eating silently through every cable. You cannot see it. You cannot feel it. But year after year, the tension holding everything together weakens, stiffens, and eventually fractures. Research in the Journal of the American Dietetic Association confirmed that dietary AGEs directly accelerate tissue aging and systemic inflammation. A separate study demonstrated this process begins accumulating measurable damage as early as your late 20s. Late 20s. When I cut refined sugar and high glycemic starches completely, something shifted in my skin within 60 days that no topical product had ever achieved. My body had simply stopped pouring rust accelerant onto its own architecture. But I had only removed 2 of the 5. The next 2, one of which I was completely convinced was a health food, were doing something far more disturbing. Not merely damaging my cells, reprogramming them at the genetic level. Genetic reprogramming is not a metaphor. It is a precise, measurable biological event. And the third food on this list was triggering it 3, sometimes 4 times a week inside my body. Not in some theoretical future. Every single time I consumed it. Processed meat, deli slices, commercial bacon, hot dogs. The preserved proteins that line every supermarket refrigerator under the quiet illusion of convenience. Here's what convenience was costing me at the cellular level. When processed meats are preserved with nitrates and cooked at high temperatures, they generate compounds called nitrosamines, molecules with a well-documented ability to directly damage DNA strands. The International Agency for Research on Cancer, operating under the World Health Organization, classified processed meat as a Group 1 carcinogen, the same category as tobacco smoke. Not because the risk magnitude is identical, but because the evidence of causation is equally unambiguous. But the carcinogen story was not what haunted me most. It was what happened in the gut. Research published in Nature Medicine revealed a pathway so elegant in its devastation it reads like biological irony. When your gut microbiome metabolizes certain compounds abundant in processed meat, it produces a molecule called TMAO, trimethylamine N-oxide. And TMAO does something deeply specific. It accelerates the stiffening and aging of your arterial walls. It does not merely raise a number on a lab panel. It physically ages your cardiovascular architecture, the very infrastructure your longevity depends on. The twist no one tells you. The damage is not just from the meat. It is from the conversation between the meat and your unique gut microbiome. 2 people can eat the same meal and age at measurably different rates because of it. I eliminated processed meat entirely, and I turned my attention to the 4th food, the one that genuinely stunned me, because I had been consuming it every single morning, convinced it was a pillar of good health. I was wrong, profoundly wrong. Commercial dairy, conventional milk, flavored yogurts, processed cheese, the foods handed to us as children as symbols of strength and growth. Here is the biological reality the marketing never mentioned. Dairy, particularly from modern commercial sources, carries a powerful hormonal signal called IGF-1. Insulin-like growth factor 1. In youth, IGF-1 is essential. It builds, it grows, it repairs. But research published in Cell Metabolism revealed something the industry did not celebrate. In adults over 50, chronically elevated IGF-1 levels, driven by high animal protein consumption, were associated with a 4-fold increase in cancer mortality and significantly accelerated biological aging markers. The mechanism runs even deeper through a cellular pathway called mTORC1, essentially the master switch between cellular growth and cellular repair. When mTORC1 is chronically activated by dairy's hormonal payload, your cells stay locked in growth mode, and a cell that never stops growing never stops to clean itself, never stops to repair itself, never activates autophagy, the body's extraordinary internal recycling system, now recognized as one of the primary engines of longevity. Commercial dairy was keeping that switch permanently jammed in the wrong position. Within 8 weeks of elimination, my hormonal panel shifted. The skin inflammation I had attributed to stress, to genetics, to everything except diet, began to resolve. My energy stopped swinging in the violent peaks and valleys I had accepted as simply adult life. 2 foods remained. The biological repair was already measurable, but the 5th food, the one I had not yet touched, was doing damage that made everything else look subtle, and almost no one is willing to talk about it honestly. There's a conversation happening in longevity science right now that the wellness industry has spent decades carefully avoiding. Not because the research is uncertain, because it is too certain. The fifth food is alcohol. And before you reach for the familiar comfort of the exception, the glass of red wine, the resveratrol studies, the French paradox, stay with me for 90 seconds. Because what the latest epigenetic research has uncovered about alcohol and biological aging is not a matter of opinion, lifestyle choice, or cultural identity. It is molecular fact. Let us dismantle the myth first, cleanly and permanently. The resveratrol argument. The idea that red wine delivers anti-aging compounds powerful enough to offset the alcohol itself. Here's what that research actually shows. To replicate the cellular benefits observed in laboratory resveratrol studies, a human being would need to consume approximately 1,000 bottles of red wine per day. The compound is real. The dosage is a fantasy, and the alcohol wrapped around it is not. What alcohol actually does, at the level where aging is decided, was documented with extraordinary precision in a landmark study published in Nature Communications. Researchers found that alcohol consumption causes measurable, dose-dependent acceleration of epigenetic aging, meaning it does not merely damage cells, it reprograms the biological clock embedded within them, pushing your DNA methylation patterns toward older, more deteriorated states. Chronological age becomes a polite fiction. Your cells are aging faster than time itself is moving. And then there's the brain. A separate Nature Communications study, tracking over 25,000 individuals, found that even moderate alcohol consumption was associated with significant reductions in brain volume and accelerated neurological aging. Not heavy drinking, not addiction, moderate. The level most people consider responsible. But the mechanism that disturbed me most was quieter than any of these. Alcohol systematically dismantles the tight junction proteins of your intestinal epithelial barrier, the microscopic cellular wall separating your gut contents from your bloodstream. As that barrier degrades, fragments of bacterial cell walls, called lipopolysaccharides, begin leaking into systemic circulation. Your immune system, encountering these fragments, responds with precisely the kind of chronic, low-grade, whole-body inflammation that researchers now identify as inflammaging, the single most well-documented driver of accelerated aging across every major organ system. Your gut was a fortress. Alcohol was quietly dismantling the walls, and the fire that got in never fully went out. I stopped completely. Not gradually, not strategically, completely. What followed was not what I expected. Within 3 weeks, my sleep architecture transformed in ways I had no framework to understand until I studied the data. Deep, slow-wave sleep, the phase during which your brain clears neurotoxic waste and your body executes its most profound cellular repair, increased by measurable degrees. I had not understood how catastrophically alcohol had been suppressing it, because the sedation had always felt like sleep. It was not sleep. It was suppression dressed as rest. My skin changed. My cognitive retrieval sharpened. The low-grade anxiety I had carried so long I believed it was simply my personality began, quietly, to dissolve. My physician repeated my biological age panel at the 18-month mark. She set down her pen. She looked at the numbers, then at me, then at the numbers again. The clock, it turned out, was not broken. It had simply been waiting for permission to run in the other direction. Here's what the science of epigenetic reprogramming now confirms, and this may be the most profound biological discovery of the last 2 decades. The damage is not permanent. The methylation patterns that alcohol, sugar, oxidized oils, processed meat, and commercial dairy had been corrupting across my DNA were not fixed sentences. They were pencil marks, and the body, given the right conditions, knows exactly how to erase them. A landmark clinical trial published in Aging Cell demonstrated that targeted lifestyle intervention produced a measurable 2.5-year reversal in biological age within 12 months. Not a slowing, a reversal. The clock did not just stop, it moved backward. The mechanism behind that reversal has a name: autophagy, the cellular self-cleaning process that mTORC1 overactivation had been suppressing for years. When the 5 foods were removed, when the chronic inflammatory signals, the glycation cascades, the hormonal dysregulation, and the epigenetic interference were systematically eliminated, autophagy reactivated. My cells began doing what they were always designed to do: dismantle the damaged, rebuild the functional, restore what decades of dietary noise had been drowning out. The practical path is not complicated, it is simply deliberate.